Today's presentation is on what is DNA. This is from the Kokomo Howard County Public Library, and I'm Ron Tiedrich, genealogy assistant. You may ask, do I really need to figure out how to use things like chromosome browsers and centimorgans to be successful in my family history? The answer is no. The basic process for figuring out your connection to a DNA cousin is to compare the total amount of DNA you share with each other, factor in things like age and even gender, and calculate how far back you would need to go to find your common ancestor. The goal is to narrow down your list of ancestor options. The more people you add to the hunt, the easier it is to figure out relationships. However, this process will never be able to add a particular name for a missing ancestor or provide ultimate definite proof of a connection. Trapped within your DNA are the stories of your ancestors. This is from Blaine Bettinger, The Family Tree Guide to DNA Testing and Genetic Genealogy. Everything you are, physically and mentally, is connected to a combination of all that has ever happened in your ancestry. What is DNA? DNA is an acronym for deoxyribonucleic acid, a hereditary material in humans and almost all organisms. A normal human cell has 92 long molecules of DNA that pair up to form 46 double-stranded chromosomes. Each chromosome forms a chromosome pair with another similar but not identical chromosome to create 23 different chromosome pairs. A human cell, as pictured here, has a cell wall, which is the outer orange, kind of a gold color, called the plasma membrane, a clear fluid inside called cytoplasm, and several bodies floating in the cytoplasm. For our study, we are concerned only with the nucleus, which is the dark purple body off to the left center, and the mitochondria. Only 3 are shown in this slide, but there are thousands inside of each cell. Nearly every cell in a person's body has the same DNA. Most DNA is located in the nucleus of a cell, or nuclear DNA. But a small amount of DNA can also be found in the mitochondria, or this is the mitochondrial DNA, or mtDNA as an abbreviation. Mitochondria are structures within cells that convert the energy from food into a form that cells can use. 4 types of DNA are used in genetic genealogy. Autosomal DNA, that's the AT and then DNA, mitochondrial DNA with the mtDNA, and the xDNA and the y-DNA, which are the female and male sex cells. The information in DNA is stored as a code made up of 4 chemical bases: adenine, which is the A, guanine, G, cytosine, C, and thymine, T. Human DNA consists of about 3 billion bases, and more than 99% of these bases are the same in all people. The order or the sequence of these bases determines the information available for building and maintaining an organism, similar to the way in which letters of the alphabet appear in a certain order to form words and sentences. DNA bases pair up with each other in a strict order: A with T, C with G, to form units called base pairs. Each base pair is also attached to a sugar molecule and a phosphate molecule. So a base plus a sugar plus a phosphate is called a nucleotide. I know this is a lot of vocabulary. You may want to look at these sometimes to get all the things straight. The figure on the left is a diagram of a short stretch of a DNA molecule. which is unwound and flattened for clarity. The boxed area at the lower left that has the letters P, S, and G encloses one nucleotide. Each nucleotide is itself made up of 3 subunits: a phosphate group, a phosphorus atom surrounded by 4 oxygen atoms. This is labeled P, over on the far left in the dark. A 5-carbon sugar called deoxyribose, labeled S, and one of 4 nitrogen-containing molecules called nucleotides, labeled A, T, C, or G. Note that in this example here, the G base is connected to a C base to the right, because C always goes with G, A goes with T. They never change that. In the figure on the right is how a DNA molecule actually looks. 2 long strands that form a spiral called a double helix. This helical ladder has the base pairs in the center, or like the rungs on a ladder, and the 5-carbon sugar and the phosphate group are the vertical sides of the ladder. This picture shows the double helix ladder of a molecule with the A and the T and the C and G base pairs surrounded by the ladder rails of sugar and phosphates. Mitochondria, the 3 red capsules in this diagram, are structures within cells that convert the energy from food into form of energy that the cells can use, which are located in the cytoplasm around the nucleus. Mitochondrial DNA spans about 16,500 DNA building blocks. These are the base pairs of over 3 billion pairs, representing a small fraction of the total DNA in each cell. Mitochondrial DNA contains 37 genes, all of which are essential for normal mitochondrial function. 13 of these genes provide instruction for making enzymes involved in oxidative phosphorylation, a process that uses oxygen and simple sugars to create adenosine triphosphate, ATP, the cell's main energy source. The remaining 24 genes provide instructions for making molecules called transfer RNA, or the T, lower T, RNA, and ribosomal RNA, rRNA, which are chemical cousins of DNA. These types of RNA help assemble protein building blocks, such as amino acids, into functioning proteins. For example, some that you may have heard of: hemoglobin, insulin, enzymes, keratin, amino acids, and there are a few more. The Human Protein Atlas, Human Protein Atlas is a Swedish-based program started in 2003 with the aim to map all the human proteins in cells, tissues, and organs. An important property of DNA is that it can replicate or make copies of itself. Each strand of DNA in the double helix can serve as a pattern for duplicating the sequence of bases. This is critical when cells divide because each new cell needs to have an exact copy of the DNA present in the parent cell. What is a gene? A gene is the basic physical and functional unit of heredity. Genes are made up of DNA. Some genes act as instructions to make molecules called proteins. However, many genes do not code for proteins. In humans, genes vary in size from a few hundred DNA bases to more than 2 million bases. The Human Genome Project estimated that humans have between 20,000 and 25,000 genes. This diagram shows 2 gene segments that are placed next to each other on a specific chromosome. And that's the chromosome, see gene 1 and gene 2 from this chromosome on the left. Every person has 2 copies of each gene, one inherited from each parent. Most genes are the same in all people, but a small number of genes, less than 1% of the total, are slightly different between people. Alleles are forms of the same gene with small differences in their sequence of DNA bases. These small differences contribute to each person's unique physical features. An allele is one of the possible forms of a gene. Most genes have two alleles, a dominant allele and a recessive allele. Therefore, a gene in a particular place or locus, that's the Latin word for place, on your DNA that controls a specific trait. In the following diagram are two alleles, each with specific chromosomes from the father, paternal, capital A and capital B, and from the mother, maternal, lowercase a and lowercase b. One set is dominant and the other set is recessive. For example, if both parents have brown eyes, and these are the dominant genes that the offspring inherits, then the offspring will have brown eyes. If the recessive genes are inherited, then the offspring may have hazel or green eyes. If the dominant genes are for blue eyes, and the offspring inherits these dominant blue genes, then the offspring will have blue eyes. Scientists keep track of genes by giving them unique names. Because gene names can be very long, genes are also assigned symbols, which are short combinations of letters and sometimes numbers that represent an abbreviated version of the gene name. For example, a gene on chromosome 7 that has been associated with cystic fibrosis is called the cystic fibrosis transmembrane conductance regulator. Its symbol is CFTR for each of the words. PIK3CA mutation is the most commonly mutated gene in HR+ or the HER2, which is the metastasized breast cancer. Female breast cancer and occurs in 40% of all women. If it advances to stage 3, it begins to metastasize. That's the MBC, and spreads into other organs and bones. What is chromosome? In the nucleus of each cell, the DNA molecule is packaged into thread-like structures called chromosomes. Each chromosome is made up of the DNA tightly coiled many times around proteins called histones that support the structure. Chromosomes are not visible in the cell's nucleus. However, during cell division, mitosis in most cells, meiosis in sex cells, the X and the Y, the bundles of chromosomes become even more tightly packed and become visible. Each chromosome has a constriction point called the centromere, which divides the chromosome into two sections or arms. The short arm is labeled the P arm. The long is labeled the Q arm. The location of the centromere on each chromosome gives the chromosome its characteristic shape and can be used to help describe the location of specific genes. A karyotype is simply a picture of a person's chromosomes. In order to get this picture, the chromosomes are isolated, stained, and examined under the microscope. The chrome part is Greek for color, and SOME is the Greek part for body. Most often, this is done using the chromosomes in the white blood cells. A picture of the chromosome is taken through the microscope. Then the picture of the chromosomes is cut up and rearranged by the chromosome's size. The chromosomes are lined up from largest to smallest. A trained cytogeneticist can look for missing or extra pieces of chromosome. This picture shows greatly enlarged structures of the 23 pairs of human chromosomes. Notice that they do have a kind of a center in some of them. Some of them don't look like they are. They're like number 4 looks kind of like almost like they're parallel, but or 13 and 14 and 15 don't really touch each other. X and the Y on the side are the X for female and Y for male. 22 pairs are called autosomal, and the 23rd pair are the allosomes. which are the X female and the Y male. A female has two X chromosomes that produce a female offspring, one from her mother and one from her father. A male has one X chromosome and one Y chromosome. His X chromosome came from his mother. The Y chromosome descends through each male and varies very little over several generations. So all the males, great-grandfather to grandfather to father to you, if you're a boy, have the same Y chromosome. Sequencing of the human genome has provided a great deal of information about each of the chromosomes. This is a table compiling statistics for the chromosomes. Each adult cell has 46 pairs of these 23 chromosomes before division. Now notice we only have 23 listed because that's only half from the father, half from the mother. 23 pairs came from the mother and 23 from the father. Note that the autosomal chromosome, that's 1 through 22, in column 2, have several hundreds or thousands of total basic pairs of genes, which are listed in the third column. The X chromosome has approximately 800 genes, and the Y chromosome has only about 50 genes. The third column shows the approximate total number of base pairs, which is about 3,079,843,747 to be exact. It is almost beyond our imagination to realize how intricately made we are. All of this is in each one of our billions of cells. The blue line, which is on the top, that's the Y DNA from the father, from your grandfather, great-grandfather, all the way back, shows the descent of the chromosome from father to son. The red line, which is at the bottom of the diagram, shows the descent of the mitochondrial DNA from the mother to all of her children, but only a daughter passes it on to each of her children. Each person received mitochondrial DNA from his or her mother, but only the daughters pass it down. Autosomal DNA, that's the orange part in the center of the diagram, the 22 pairs in slide 27, descends from all your ancestors, represented by the orange lines. Genetic genealogy is the use of DNA testing in combination with traditional genealogical methods, or the paper trail, to infer relationships between individuals and to find ancestors. Genetic genealogy involves the use of genealogical DNA testing to determine the level and the type of the genetic relationship between individuals. This application of genetics became popular with family historians in the first part of the 21st century as tests became more affordable. The tests have been promoted by amateur groups such as surname study groups or regional genealogical groups, as well as research projects such as the following. The Genographic Project, launched on April the 13th, 2005 by the National Geographic Society, which is a genetic anthropological study that aims to map historical family migration patterns by collecting and analyzing DNA samples. The current phase of this project is called Geno 2.0, Next Generation. As of 2019, 26 million people have been tested in over 140 countries. As this field has developed, the aims of practitioners broadened, with many seeking knowledge of their ancestry beyond the most recent centuries for which traditional pedigrees can be constructed. This project does not track individual genealogical ancestries, but rather ethnic groups. The U.S. is a multiracial and multi-ethnic country. The state officially categorizes its population into 6 ethnic groups: white, African American, Native American or Alaskan Native, Pacific Islander or Asian, and Native Hawaiian. The investigation of surnames in genetics began with George Darwin, a son of the famous Charles Darwin. In 1875, George Darwin used surnames to estimate the frequency of first cousin marriages. This was in the United Kingdom. To estimate those and to calculate the expected incidence of marriages between people of the same surname. This is called isonomy. Iso is the Greek for same and onym is for name. He arrived at a figure between 2.25% and 4.5% for cousin marriages in the population of Great Britain, higher among the upper classes and lower among the general rural population. Upper classes tend to intermarry mainly to maintain control over land and political power. In lower classes, first cousin marriages are rarer, but do occur in certain religious groups who tend to marry within a particular sect. Now, how many ancestors do you have? You have 2 parents, 4 grandparents, 8 great-grandparents, and 16 great-great-grandparents. Notice that the number continues to double as you go back in time. 11 generations in this diagram, 1,024 different people at the bottom of it. In 20 generations, you would have 1,048,576 individual ancestors. If you were to travel back 35 generations, you would have 68,719,476,734 different ancestors. And everyone alive today in the entire population of the world would have exactly the same set of common ancestors, although they would be related through different routes. Since this mathematical conundrum is impossible, the result is called pedigree collapse, showing that at more than one point in the last 500 years, some of your ancestors were closely related to one another. A dramatic example of pedigree collapse can be found in this ancestry of Charles II of Spain, who lived from 1661 to 1700. He's at the bottom of the chart. You work your way back up. He was the last of the Habsburg rulers of the Spanish Empire. He had 2 parents, but they were closely related. His mother was his father's niece, as were his grandparents and great-grandparents. His only 8th great-grandparents were Philip I of Castile, the Duke of Burgundy, 1478 to 1506, and Joanna of Castile, 1479 to 1455. The marriage was one of a set of family alliances between the Habsburgs, the Holy Roman Emperors, and the Trastamaraes, an ancient Spanish dynasty, designed to strengthen both families against the growing French power, which geographically lay between the countries of Burgundy to the north and Spain to the south. Instead of 256 individuals in the 8th generation, where you see Philip and Joanna, Charles had only 2. Another example is the ancestry of Engelbertha Harmonia Roydink, whose great-great-grandparents should number 16 individuals, but are only 6 individuals, a collapse of 62.5%. She descends from her great-great-grandparents, Jan and Clasina, 4 different ways. She herself married Johann Gerhard Roydink, who was her first cousin. Now, what is the Human Genome Project? The Human Genome Project is an international scientific research project with the goal of determining the sequence of nucleotide base pairs, that is the A with a T or the C with a G, that make up human DNA, and of identifying and mapping all the genes of the human genome from both a physical and a functional standpoint. It remains the world's largest collaborative biological project. After the idea was picked up in 1984 by the US government, when the planning started, the project formally launched in 1990 and was declared complete on April the 14th, 2003. The Human Genome Project originally aimed to map the nucleotides contained in a human haploid reference genome, more than 3 billion, if you remember that big number. The genome of any given individual is unique. Mapping the human genome involves sequencing a small number of individuals and then assembling these together to get a complete sequence for each chromosome. Therefore, the finished human genome is a mosaic, not representing all of any one individual. Two conclusions are of genealogical importance. One, there are approximately 22,300 protein coding genes in human beings, the same range as in other mammals. And two, the human genome has significantly nearly identical repeated sections of DNA and more segmental duplications than had been previously suspected. Of course, there are some ethical, moral, and health issues. At the onset of the Human Genome Project, several ethical, legal, and social concerns were raised regarding how increased knowledge of the human genome could be used to discriminate against people. One of the main concerns of most individuals was the fear that both employees and health insurance companies would refuse to hire individuals or refuse to provide insurance to people because of a health concern indicated by someone's genes. In 1996, the United States passed the Health Insurance Portability and Accountability Act, which we refer to as HIPAA, which protects against the unauthorized and non-consensual release of individually identifiable health information to any entity not actively engaged in the provision of healthcare services to a patient. That is, it does not go to employers or to insurance companies. Other nations have not passed such protections. Along with identifying all of the approximately 20,000 to 25,000 genes in the human genome, the Human Genome Project also sought to address the ethical, legal, and social issues which were created by the onset of the project. For that, the Ethical, Legal, and Social Implications Program was founded in 1990. 5% of the annual budget was allocated to address the ELSI arising from the project. This budget started at approximately $101.57 million in the year 1990, but increased to approximately $18 million in the year 2014, for which the last year we have any figures. While the project may offer significant benefits to medicine and science research, some authors have emphasized the need to address the potential social consequences of mapping the human genome. Molecularizing diseases and their possible cures will have a profound impact on what patients expect from medical help and the new generation of doctors' perceptions of illness. Is it ethically moral to remove a defective gene and replace it with a healthy one? Who has the authority to initiate such a procedure? What effects will such a procedure have on future generations? All right. What is epigenetics? DNA modifications that do not change the DNA sequence can affect gene activity. Chemical compounds that are added to single genes can regulate their activity. These modifications are known as epigenetic changes. The epigenome comprises all of the chemical compounds that have been added to the entirety of one's DNA, the genome, as a way to regulate the activity or the expression of all the genes within the genome. The chemical compounds of the epigenome are not part of the DNA sequence, but only are attached to DNA. Epigenetic modifications remain as cells divide, and in some cases can be inherited through the generations. Environmental influences, such as a person's diet and exposure to pollutants, can also impact the epigenome. Epigenetic changes can help determine whether genes are turned on or off and can influence the production of proteins in certain cells. ensuring that only necessary proteins are produced. A GMO, you've probably heard that in television on commercials, or a genetically modified organism is a plant, animal, microorganism, or any organism whose genetic makeup has been modified in a laboratory using genetic engineering or transgenic technology. This creates combinations of plant, animal, bacterial, and virus genes that do not occur in nature or through traditional crossbreeding methods. Genetic modification affects many of the products we consume on a daily basis. As the number of GMOs available for commercial use grows every year, the Non-GMO Project works diligently to provide the most accurate, up-to-date standards for non-GMO verification. Errors in the epigenetic process, such as modifying the wrong gene or failing to add a compound to a gene, can lead to abnormal gene activity or inactivity and can cause genetic disorders. Some of these include cancers, metabolic disorders, and degenerative disorders. The jury is still out on the possibilities epigenetics will have on eradicating disorders and diseases to create the perfect human being. This is an issue that sounds like science fiction, but it is gradually changing our concept of diseases that have plagued mankind from the beginning of time. For example, CCR5 delta 32 is a mutation that saved your ancestors from the bubonic plague in the 14th century. Otherwise, you would not be alive today. Centimorgan, which is usually the cm, is a unit of measure for about half. The centimorgan decreases by about half as one goes back in each generation. So a person that could share about 25% with grandparent and only 12.25% with any first cousin. With fourth cousin, the shared number drops to about 0.27. With a sixth cousin, you would share no more than 0.01%. As you can see, this is not an exact relationship, since the amount of autosomal DNA inherited from each preceding generation decreases dramatically. A known paternal third cousin and I share some DNA, but Ancestry DNA claims that we are fourth cousin, not third cousin. You must decide for yourself whether to take such a test to find long-term or long-lost cousins. The only true relationship must be verified in a paper trail by documenting each place on the pedigree chart with primary source materials. Most DNA testing services will include the number of centimorgans in matching DNA segments. You do not have to carry out any complicated calculations. After the results are determined, go to your results page in your sequence account and check whether there are any matches. In an Ancestry DNA, for example, you can click on a specific match and then click on or hover on the small I next to the confidence label. This brings up a box showing the number of centimorgans or shared segments. You can convert the centimorgans into percentages simply by dividing the total number of centimorgans by 68, since there are roughly 6,800 possible centimorgans in a person's DNA. So if you share 3,400 centimorgans with a match, you can say you share 50% of your DNA with that person. A 50% match is likely a parent or a child, while a 1% match is likely a third cousin, because remember, it reduces half by the time you go back a generation or remove yourself from yourself. If you are searching for an unidentified parent, this could be a lucky strike. This is probably more than you really need to know about DNA and finding genetic matches with an autosomal DNA test. The companies already mentioned have done the mathematical calculations for you. All you need to do is check the results and contact for the close matches. Who am I? Where do I come from? What's in my DNA determines what I look like, what habits I have, what choices I make, whom I will become. What am I passing on to my children? These are age-old questions we have always asked ourselves. Stay tuned. Genetic genealogy is just beginning to answer some of these questions. The answers will change tomorrow and the day after that. Aren't you glad that you are alive? Thank you for listening.